metabolomics, I believe, is the future of drug target and everything. And so this conference that you hold every year is really very important. So I would like to talk today about a nutritional factor, a micronutrient, vitamin B12. And this is specifically relevant for India. That is because Indian population is thought to be deficient in this vitamin. So about 30 to 40% of people have low levels of these vitamin. And that is predominantly because of low intake, because this is one vitamin which is completely absent in plant products, and 29% of Indians are known to be vegetarians. I'm having some issue with... Okay. So the second problem is malabsorption. So vitamin B12, actually, when when you take in vitamin B12, it is transported by 3 different proteins. And one of the protein is intrinsic factor. Now, Indians, almost 50 to 90%, depending upon the region, have H. pylori infection. Now, what H. pylori does is it inhibits intrinsic factor, and thereby the transport of B12 is inhibited. Then there are genetic factors which lead to defective transport. So, for example, we had shown earlier that fucosyl transferase polymorphism results in low vitamin B12 levels. Further, there are other diseases. So you take drugs, so they can actually limit the availability of B12. One such drug is, metformin, which is often given for diabetes. And people with prolonged intake of metformin have low B12. And we had earlier shown that, Indians have very low levels of HDL, and that is one contrasting factor of Indian population as against the world population, where they have high LDL, but their HDL level is normal. In India, it is the reverse. Here, the HDL level, which is often called the good cholesterol, is low. And we had shown that, HDL has direct correlation with vitamin B12 levels. So what we wanted to do is to understand if vitamin B12 levels have any influence on the lipids. And for that, we, in collaboration with CCMB and IICB, had developed a maternal B12 deficient rat model and wanted to look at their offspring. We did this because in India, it is shown that, during pregnancy, the mothers are very highly deficient in vitamin B12. So almost anywhere between 50 to 70% of pregnant women are deficient in B12. And we wanted to validate the Barker's hypothesis, which clearly states that in utero nutritional experiences could influence the offspring towards the risk factors for obesity, and this could be due to epigenetic mechanisms. So what we did was, we took female Vista rats and fed them with B12 deficient diet for 3 months. And then after 3 months, we mated them with normal males with adequate B12 levels. A subset of these females, what we did was, we before mating, we supplemented them with B12 to see that if we supplement B12 at conception, is the whatever effect we are seeing, are they reversed or not? So after 3 months of B12 deficiency, we got that the mothers were about 36% deficient in B12, which is a moderate deficiency, which is what we usually find in the country. So we then looked at the offspring, and we found that because of the offspring, although the mothers were about 35% deficient, the offspring males were around 70% deficient, the females were about 60-65% deficient. And as expected the homocysteine levels were high in both males and females. But what was interesting that cysteine levels were only high in males, but there was no alteration in cysteine levels in females. Also, we looked at the total fat. You can see the body weight was not changed that much. Lean fat mass was also not varying, but total fat content was high only in the male offspring born to vitamin B12 deficient mothers, but there was no such change in female offsprings. This was very interesting because one particular nutrient, if it is deficient in mothers, it is showing that it is leading to two different phenotypes in the offspring, and which is sex dependent. We also then looked at HDL and triglycerides. The others were more or less same both in males and females, but HDL was significantly low in males. Nothing happened in females. Triglyceride levels were significantly high in males. Again, nothing happened in females. So this was really an interesting thing. So what we did was, like Alice was talking about 5P, in this case, we used all sorts of omics to try and understand what are the pathways that are being altered. So for the first time, we did a DNA methylation in rat, and these are all data from rat liver, because liver is the metabolic hub of all the organs. And we found that, in males, if you can see, there is a hypermethylation near the transcription start site, and the blue one is the controls who had adequate B12. And the gray one, you can see, is the ones where we had supplemented back B12 during conception. So you can see that it is slightly, recovering from the orange bar. It has come down towards the blue bar. While in female, we did not see any such effect. And when we looked at the differentially methylated regions, so we did a MEDIP sequencing here, we could see much more regions altered in males as compared to females. And the overlap was at best 10%. So we concluded that there is a sexually dimorphic alteration in DNA methylation, higher dysregulation of methylation in the vitamin B12 restricted group, and repletion at conception partially reverted methylation signatures back to the normal levels. And when we looked at the pathways, we found that pathways leading regulating glucose, lipid metabolism, insulin sensitivity, etc., were being altered. We also did the proteomics of the same liver tissue, and here also you can see that the proteomic signature of the males and the females were different. Again, we see only 10% overlap between males and females. And when we compared the data from proteomics and DNA methylation, one thing that stood out is the PPR signaling pathway. PPR is a transcription factor that actually regulates carbohydrate metabolism, amino acid metabolism, and lipid metabolism. And PPR alpha gene itself showed hypermethylation in the promoter region. And when we looked at the protein levels, we could see that in females, actually the levels of PPR alpha increased, but there was a significant decrease of PPR alpha in males. And this, we had earlier published that when there is very severe deficiency, PPR signaling pathway is altered. So because it was controlling carbohydrate, amino acid, and lipid, we tried to look at the metabolites of glycolysis pathway. We looked at the metabolites of TCA cycle, and these were all, relative quantitation done using targeted metabolomics. And if you look at the first lane and the third lane, these are males and females. You can see that vitamin B12 restriction had distinct changes in males, but there was very little alteration in females. And the second one here is from the animals where we had supplemented B12 at conception, and we can see that some of the signatures are coming back, which were altered in the vitamin B12 restricted males. We also looked at the 1-carbon metabolism pathway. Again, the first column, you can see all of these were upregulated, but after giving them B12 at conception, most of them reverted back. In females, the signatures were completely different. And the bottom one, I'll come to it later again. These are the steroid, and I'll come to it later. On the right hand side, you can see the amino acid profiles. All the amino acids were more or less altered in vitamin B12 restricted males, which reverted back closer to normal when you supplemented them at conception. But in females, the signature was more or less different than what you see in males. So clearly, using DNA methylation, proteomics, and metabolomics, we see distinct signatures between males and female offsprings that are born to the same B12 deficient mothers. We then tried to understand what are there changes in lipids. So what we did here was we developed a method where we could identify more than 1000 lipid species in a 24-minute run of mass spec, and we found that about 883 lipid species from 17 lipid classes came up with a CV of less than 30%, which is considered to be very good in this case. And using this approach, what we found that when we took the liver tissue, the cholesterol ester was significantly high in males, but there was no such effect in females. Cholesterol level, you can see that it was more than twofold increased in males, but not in females. Triglyceride levels were significantly low in the tissue. We have seen that in the plasma it is high, but in the tissue, triglyceride levels were lower as compared to that of females. So clearly distinct signatures, and clearly there is an accumulation of cholesterol. And if there is an accumulation of cholesterol, we expect changes in the steroid hormone synthesis, and we individually checked all these metabolites in the steroid hormone metabolism, and you can see that most of them are pretty high in males. In females also they are high, but they are not that high. For example, here you can see corticosterone is 3-fold upregulated in male, but it is only 1.1-fold upregulated in females. So I'm sorry, the pink one is females and the blue one is males. Now, so we can, and this is the heat map where you can see all the steroid hormones are upregulated in males, but not that much in females. So what we also found out, one interesting thing, that is there is an enzyme called SALT1A1 which converts estrone to estrone sulfate, thereby degrading the estrogen pathway. So estrogen consists of estrone, estradiol, and estriol. So if this is hypermethylated, this is low, then you will expect more of estradiol, estriol, and very less amount of estrone sulfate. So this was very interesting, and what we found was in males and females, you can see that this is control, and these are vitamin B12 restricted females. You can see this almost absent. Even in males, they are very low as compared to the controls. Now, one interesting observation here is that there are reports that there is a sex-specific effect of estrogen sulfotransferase on mouse model of type 2 diabetes. So loss of EST in female, which actually what is that we are finding, improved metabolic function in OB mice, but the effect of EST was sex-specific, and EST ablation in OB males actually exacerbated the diabetic phenotype. So the same protein, if it is low, it actually is beneficial for females, but it is detrimental in males. And one of the reasons could be increased in estradiol. And there are reports suggesting that when estradiol is high, it decreases taurine level by reducing cysteine sulfonic acid. Now we found that estrogen sulfotransferase is low, so estradiol is high, and then there should be some effect on cysteine sulfonic acid, thereby taurine. We wanted to check that. So we had already found that the homocysteine was high in both males and females. Cystathionine beta synthase was high in males but low in females. That's why cysteine was only high in males, not in females. And so what we showed that EST is low in females, you have estradiol high, and that's why you're supposed to get cysteine sulfonic acid low, and that is exactly what you can see here. Cysteine sulfonic acid decarboxylase is very low in females, but no such effect is observed in males. And if cysteine sulfonic acid is low, you will have low taurine levels, and we actually found low taurine levels in females, but not in males. So what is interesting is CSIR is actually considered as a rate-limiting step for taurine biosynthesis, and downregulation of CSIR in female hints at impaired osteoblast function. So in males, we had found that atherogenic risk factors like triglyceride, uh, HDL, etc., to be altered, but in females, we are seeing more of taurine kind of thing to be altered. This is actually shown here. I would not, this is summarizing it. So this is what happens in males. This is what happens in females. So in males, you get more atherogenic risk, while in females, you get more of bone density loss. And that is exactly what the phenotype that we find in our country. But the issue was, all we have done is in rat, right? So other than fatty acid metabolism, other functions of PPR alpha are actually specific to rodents. Because mice and rats, they do not have CETP, that is cholesterol ester transfer protein. And often in case of these lipidomic signatures, rat is not a good mimic for human. So after finding the pathways that are altered in rats, we also tried to understand what happens in humans. And for this, we used Phenom India study, which is a large longitudinal cohort that is being undertaken in CSIR for the last couple of years. So here what is being done is CSIR employees, pensioners, and their spouse are being enrolled on voluntary basis. The idea of this is, like Alice pointed out, 5P. The idea of this is to develop clinically useful personalized risk prediction scores for basically for non-communicable diseases. Why CSIR? One of the biggest reason is CSIR is a urban and semi-urban population spread across the country. It has already established network of scientific and research laboratories and is expected to have high enthusiasm. And it has excellent in-house capacities for conducting clinical omics study and big data analysis. So what we thought was, as a pilot for the entire country, if we can do Phenom India for CSIR people, and later more and more people can join in, and thereby we can collect a lot of data. So here what we are doing is, we are doing lung function tests, we are doing liver fibroscan, skin testing, body composition. It's just not weight, but we are looking at the fat mass distribution. We are doing ECG, we are doing blood biochemistry and different molecular analysis, including proteomics, metabolomics, genomics, etc. And the data is being stored at a data repository, and samples have been stored in a biorepository, which was actually inaugurated a couple of months back by Honorable Minister Dr. Jitendra Singh. So these are the things that we are doing. We are doing proteomics, metabolomics, extended biochemistry. We are looking at cytokines. We are looking at telomere length. We are doing genomics. We are for the first time doing metabolomics. So we are taking about 32 elements. We are looking at the plasma levels in humans. We are doing microRNA. And as Alice mentioned, we have already completed 6,000 microbiome from these people. So if we have to look at this, so generally, using this instrument, the normal level of B12 is considered to be 280. You can find that males, the median level is only 296. So in general, Indian population is low. 347 is the female, and if you take overall, it's about 317 milligram per ml. And so the next question we asked was, like in rats, does B12 deficiency in human also alter lipid profile? And for this, we use the biocrates. So we have currently done around 5,000 samples in using biocrates, and we are also following up these patients. So in next year, again, we'll run a similar number of samples using biocrates. And I don't want to talk about this. These people who have been using this are familiar with it. So what we did here in this case, what is interesting is we divided the assay such that 3 different sites, 3 different labs in 3 different parts of the country did the biocrate assay. It was done in more than 1,000 samples in all the 3 centers. And what was very interesting and heartening to see that the results were very much concordant. Although we tried to normalize it, but it was very little normalization was required. So we had about when I did this analysis, we had almost 4,000 samples. We excluded 2,400 samples because we wanted to look at the pure population. Anybody who was taking any drugs like metformin or they were taking supplements, we excluded them. We had 1,470 pure samples who had no drug history or no medicines they take. And we also removed metabolites that are present in 80% of the sample. So we got about 446 metabolites quantified, and these 446 metabolites were there found in all three centers. And this is just a demographic status we expected. So what we did was we divided the 1,470 people in different groups. People who have less than 200 B12, 200 to 300, 300 to 400, and greater than 400. So we wanted to see whether we get any graded effect. The vegetarian population were higher in B12 deficient group as expected. So let's start with HDL and ApoA1. You can see here that the blue bar here is the low B12, and as B12 level increases, apolipoprotein A1 increases. Similar is the case of HDL. As your B12 level increase, HDL increase. You will appreciate that the same thing we observed in case of rats also. I will quickly go through this. I just don't want to spend too much time. There are differences in amino acid level. You can see proline significantly decreasing while a B12 level is increasing. And if you have high proline, that is one of the factors which can tell you that you have low muscle mass. We had cholesterol esters which were significantly low in B12 as the B12 levels were low, and low cholesterol ester 20:5 and 22:6 has been associated with liver cirrhosis. You can see here that these are, we have overall male and female, and if you can see, in some cases, male and female behave differently again. This was phosphatidylcholine, and phosphatidylcholine is low in low B12. As the B12 levels increase, we saw a graded increase of phosphatidylcholine. So low levels of phosphatidylcholine have also been associated with liver diseases. The final thing that I want to show you is the triglycerides. Although when we measured triglycerides using automated analyzer, there was no change. But there has been reports like this in several literature where they could not find any difference in cardiac disease, triglyceride, and controls. And that is probably because in the automated analyzer, what we measure is not triglycerol, but what we measure is dihydroxyacetone phosphate and the H2O2 that is liberated. So we are actually measuring this and indirectly linking it to triacylglycerol. And this is a great example where mass spec based studies are important for understanding the levels of different chain lengths of triglyceride. And this is one example where you can see that if you have low chain triglyceride, they are actually higher in low B12 group, but large chain triglycerides are much more higher in low B12 group. So you can see the blue one here, and it slowly decreases. But medium chain triglycerides and low chain triglycerides, which are good for the heart, are actually higher in higher B12 group. So clearly, the chain length of triglyceride is important. So just measuring triglyceride probably will not do. You need to look at the chain length. You need to look at the composition. So what we have now found, and I'm not showing you, is triglycerides which are composed of fatty acids which are omega-3 in nature. They are much more higher if your B12 levels are high. If your B12 levels are low, omega-3 linked triglycerides are much lower. So, this is why you probably need to look at the mass spec of metabolomics, and biocrates is a very good example actually because it's giving you quantitative results. This is my group.
Timekeeping.
And these are the
cohort. Timekeeping, 1 minute left, please.
Yeah, I'm done. So thank you for inviting me again, and thank you for listening.